Well, good afternoon, everyone, and welcome to the AFOSR Chief Scientist Distinguished Lecture Series. As we move forward, we have been very lucky to have some of the brightest and best professors from around the world present their work to us. And continuing in that theme of things brought to us by our program officers nationally and our international program officers. Continuing with that theme, I have the pleasure of welcoming to our lecture Dr. Professor Tiffany Walsh. I had the pleasure of meeting Tiff this last December at the University of Adelaide. I was attending the 24th Australian International Physics Conference, sponsored both by Australia and New Zealand. And then there was a conference on biotechnology taking place by a dear friend and colleague, Professor Mark Hutchison, and I was able to meet Professor Walsh, and we were able to spend an afternoon talking about the work that she's been doing on behalf of Deakin University, as well as the Department of the Air Force through our Asian Office of Research and Development in Tokyo. And with that, I want to introduce Dr. Todd Rushing, who will actually introduce Professor Walsh to us. And thank you for attending.
Thank you, Dr. Roach. Hello, good afternoon, good morning, everyone. This is Todd Rushing. I'm a program officer in the Tokyo office and in the Asian Office of Aerospace Research and Development. And it is my pleasure this morning on this side of the world to introduce Professor Tiffany "Tiff" Walsh. She is with the Institute for Frontier Materials at Deakin University in Australia. Tiff received her undergraduate in chemistry from Melbourne University in Australia. And then she went on to earn a PhD in theoretical chemistry at Cambridge in the UK. She was on a full scholarship there as a Cambridge Commonwealth Trust Scholar. And her advisor at Cambridge was Professor David Wales. After she finished her PhD, she moved to Oxford University and completed a postdoc at the Materials Modeling Laboratory, working with Professor Adrian Sutton. And at that time, she was on a Gladstone Fellowship at the Materials Modeling Laboratory in Oxford. After the postdoc, she moved to a faculty position at the University of Warwick in the UK as a joint appointment with Department of Chemistry and the Center for Scientific Computing. And there she was an assistant professor and associate professor for a while before moving to Deakin University. So currently, since 2012, she is a professor at Deakin University in the Institute for Frontier Materials. Tiff is currently working on her second grant from AORD. A few years ago, she had an AORD grant, and that topic was integration of experiment and modeling to advance biosensor design based on conformational switching of surface absorbed nucleic acids. That was a 2-year project, and she published 4 papers out of that grant. And after a little pause, she is now on a new current grant where I'm the program officer. And her current grant was just awarded a few months ago. And the topic is nanoscale biosensing devices, fundamental insight on the sensing mechanisms. And the exciting thing about this grant is it is a very close global collaboration. Her collaborator is, her academic collaborator is Professor Matteo Palma at the School of Biological and Chemical Sciences at Queen Mary University of London. And in addition to that collaboration, this grant and her work has very strong support and collaboration from AFRL. And the people involved directly at AFRL are Dr. Jorge Chavez Benavides of the Airman Systems Directorate, that's RH, and also Dr. Peter Morale of the Materials and Manufacturing Directorate, RX. So we're very excited to learn about the research that she will present this morning. So please welcome Professor Walsh.
Thank you, Todd, for that really kind introduction. And Pat, thank you so much for this invitation. It is an immense honour and pleasure to be able to present at this distinguished lecture series. And thanks to all of you for coming along. And hello, good afternoon, good morning, wherever you are in the world. What I'm going to do now is share my screen, and we can get started with the presentation. Okay, so can we see all that? I'll just put it into presentation mode. Sorry, you've got a sneak preview there of one of the slides, and we can get started. If everyone can see that, okay?
Yes.
Wonderful, thank you. So thank you for coming along today. I'm going to be telling you about some work I've done as part of another international collaboration that was funded by the AFOSR Natural Materials Program. And that's work that I've been doing with Professor Mark Knecht at the University of Miami. This is a long-standing collaboration that I have with Mark. We've worked a lot in the bio-nano area. We have done a lot of work in terms of investigating the interactions between peptides and nanoparticles. And part of the actual AO-ARD grants that Todd told you about before was really building off that work that I'd done with Mark, and then branching out into the human performance space by modeling DNA interactions with gold nanoparticles. And now with the work that I'm doing with Matteo in London on this second AO-ARD grant, we're leveraging the experience we gained off the work I'm going to tell you about today to be able to get started on that. So there's what's really nice about this is that there's great interconnections between the support and the projects that we've been able to do internationally with our partners, both academic and of course our partners here at that we work with at AFRL. And they were also a really key part of this story. So in the slides, you'll see this U here. This is the Miami University logo, and that's because Mark and I work really closely together on this. And although I'm going to be telling you about the work that I do, which is modeling and simulation of materials at the atomic and molecular level, it's so important when you do modeling and simulation that you have experimental backup, that you have an experimental partner who can basically help you keep your feet on the ground, really, and make sure that whatever that you're doing or what you're predicting is really on the right track. And so we always make it a point to work with experimental partners who can help us understand if we're doing the right thing, and that who can constantly challenge us to continue to innovate and try and find new ways to explore the phenomena that we're interested in, which is all about that interface between biomolecules and inorganic materials. So today I'm going to be telling you about work that I've been doing with Mark over the past 3 years, a bit more, in terms of understanding how we can use peptides to exfoliate and organize 2D materials. And this has been a lot of fun. But before I get into that, I want to, just because I know some people here might know where Deakin is, but maybe not everyone on the call does. So just to explain where Deakin University is in the top left-hand corner. I don't know if you can see my cursor, but so we're down here in the south of Australia, the southeast of Australia, not too far from Melbourne, about an hour's drive west out of Melbourne. Down in the bottom left here, this is my institute. So that's what it looks like. And to the right of that is the sort of local environment, and in the panel above, the coastline, pretty close to where I live. So this is where we are. It's a really wonderful place. It's beautiful. Deakin University, and in particular my institute, is very stimulating. We have a lot of, it's a materials institute, so we really do everything from aircraft down to, molecular sensors. There's a wide range of activities and a huge amount of expertise in materials. So you'd be very welcome to come and visit and check it out. It's one of the nation's leading materials institutes. But about the kind of work I do. So to start with, just to explain to you what my lab looks like. It looks like this, a machine room full of computers. And usually these computers are located very far away from where I actually work. So my research is 100% done on the computer. And we use national facilities to be able to do this research. And these national facilities, well, one's based in Canberra and the other one is based in Perth. And so I never usually go into this machine room and do anything, right? I'm only ever accessing these computers remotely. So this means I could be anywhere. I could be in Hawaii and I could do my work if I needed to. But the point is that these national supercomputing facilities have been instrumental in being able to do our research on these Air Force funded grants. And it's a substantial contribution that I'm very grateful for in our national research infrastructure to be able to do that. So they've been really key to us being able to do the work that we do. So what do I do, or what does my team do? What's our specialty? Is that we use computer simulations and computer modeling to predict the properties and the behaviors at that molecular level of these quite complex and challenging interfaces, the interface between a biomolecule or biomolecules and any kind of solid surface. And the thing about molecules on surfaces, in particular under aqueous conditions, so in the presence of liquid water, is that it's very tough to be able to use experiment to get the structural information you need at that required level of resolution. It's really tough. And so this is something where molecular simulation can be very helpful and give some insights into what those structures might look like. So that's our structural prediction. We can also get a handle on how strong a molecule binds to a surface, so whether it's strong or weak, whether that molecule has any selectivity. So does it prefer to bind to one material over another? And then we can start to pinpoint what it is about that molecule that makes those interactions so strong or weak. And then we can give guidance on how to tune up or tune down those particular interactions. So what we provide in my team is very fundamental knowledge about how these molecules behave at a molecular and atomic sort of level of resolution when they're at an interface, which is a sort of tricky situation to be able to get that same level of detailed information from an experiment. But it's really important that whatever we do in our simulations, we're able ultimately to connect back to experiment so that we can understand if our predictions and our guidance is actually making sense. So as I've said, Mark Connect and I have been doing this for some time now. We've worked together for over a decade, and we have a lot of expertise in terms of understanding how to do modeling and experiments of peptides on metal nanoparticles in particular, but all sorts of nanoparticles. We wrote a ChemRev on this in 2016, but then we've been interested in thinking about how we could be doing more with the peptide interface interactions in terms of applying these to 2D materials. One other thing I wanted to tell you about before I launch into telling you about the 2D material work is just some examples of how peptides or peptoids or biomolecules can be used to actually create materials that have really exceptional properties. And in the top panel, what I'm showing you here is some work that I did actually with Chunlong Chen and Jim Diorio, where they were using peptoids in this case, not peptides, but very simple, very similar kind of approach. They were using these peptoids and trying to explore different peptoid sequences to be able to nucleate and grow not just gold nanoparticles, but what you see here in the right hand of this top panel, these what we were calling nanogold sort of coral structures. And what I'm showing you on the left-hand side, if you're a scuba diver, you might have seen at some point, especially if you dive on the Great Barrier Reef, which is something I recommend you do. It's a lot of fun. You'll see a lot of this staghorn coral that I'm showing here on the left. And our nanogold coral structures look just like that. Originally, we called them nano hedgehog structures, and one of the reviewers of our paper said, well, hedgehog quills kind of taper down to a point, and your structures don't do that, so you can't call it a hedgehog. So we found a happy compromise in calling them a coral. But the real point about this is that this very branched nanostructure could give exceptional plasmonic properties, could confer these. And really, we did a lot of modeling work to help Jim and Chunlong and their teams understand how could they manipulate that peptide sequence to get the most reliable pathway to creating these coral structures. Similarly, in the panel at the bottom, this is work that I did with Mark Connect and our collaborators at University of Buffalo, so Paris Prasad and Mark Swihart, where we were using the concept of a chimeric peptide-based molecule and using that to try and direct the organization and activation of metallic nanoparticles. So on this bottom panel on the right-hand side, the concept here is to design a molecule that will have a domain at one end that, say, can recognize and bind to gold, but not to silver, and then have a peptide domain at the other end of the molecule that can bind to silver, but maybe won't bind so well to gold, and have an actuating linker in between those two domains, something that we can stimulate with light or heat or pH, and to be able to use this to perhaps manipulate those plasmonic properties of the material that we ended up creating. So we gained a lot of expertise in understanding binding selectivity and also the ability of these chimeric molecules to organize materials that we've since carried forward into this 2D material project. So one of the key things that if you don't work in this area that you might not be aware of is that these peptide sequences, so I'm showing you one here in the top left, this AQN, PSD, blah, 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 sequence. So that's a sequence that's known to bind to gallium arsenide, and it binds selectively versus binding onto, say, silica. We know that if we just take this sequence and we scramble it, so we mix it up, and you'll get the sequence here on the right, that can completely destroy its binding affinity and its binding selectivity for its target material. So we know that actually the sequence really matters. And therefore, we need to try and understand why it is that the sequence, as I've got here on the left-hand side, can confer the properties that it does. And what's really the key are these missing bits in the gray, which are hard sometimes to determine. So the sequence will confer a 3D structure of this molecule when it's sitting at the interface. That 3D structure really can dictate that surface contact, or there's a lot of interplay between the 3D structure and the modes of contact to that molecule on the surface. And therefore, that's how you get the properties. And these gray items, the 3D structures at the surface and how it makes surface contact, very tricky to be able to resolve using experimental techniques. And here I'm showing you some really great pioneering work that was done at AFRL with Peter Moreau and the rest of the team here, Rajesh Naik. And here you can see that they were able to resolve some structural information of peptides binding onto titania. And this was really an incredible breakthrough using NMR. And this kind of experimental information is so precious for modelers like me to be able to use and understand and compare against. But the thing is, it's very challenging to obtain, and it would be great if there was more of this information. But to date, this is, I guess, proved very challenging, and it's something that we would always like to welcome more of in the simulation world. So the point is that the molecular simulations we do can help resolve this kind of nexus and help identify these 3D structures and potential ways of that molecule making surface contact. So it can start connecting the dots along this sort of pathway of our understanding. As a modeler, there's all these things that I'd like to do that are really kind of idealistic. So the thing about being a modeler is that you can always produce something, right? You can always produce data. That's not the hard part. The hard part is really producing relevant data and trying to make sure that you're not overinterpreting it. So when you do molecular simulation, you've got this dilemma. You want to be rigorous, but you also want to be relevant. And these two aspirations kind of put you into conflict. So in terms of rigor, I want to do things in the most realistic way I can, and I want to be able to make sure I sample all the conformations of the molecule, and I want to make sure that the way I describe the interactions between the atoms in my simulation is the best possible. But I live also in the real world, and for me to be able to stay rigorous, I need to make sure that I can model a system that has all the right details in it, right? So, I've got to model the surface correctly, and it might be all kind of messed up and messy. And I'll talk a bit about that later in terms of how we do that. And it's simply, for example, I can't exhaustively sample the entire conformational space of the biomolecule. It's not possible. So I have to be really targeted about that. And also the way I describe my interatomic interactions is always going to have approximations, because in our simulations, we might have 1 million atoms. If you've got 1 million atoms, you can't start getting all high and mighty about how sort of super accurate your interatomic interactions are. You've got to be able to approximate them somehow. You've got to understand, well, what approximations can I live with here, and which ones is it okay for me to feel a bit more relaxed about? It's understanding those boundaries that's really key to doing modeling work. Some of the work we do involves being able to describe or capture the conformational states of these peptides. And I should have mentioned before, when I talked about the experimental work, that these peptides are typically intrinsically disordered, which means they don't always adopt those sort of nice secondary structures that you learned about maybe in first year university or whatever. So they don't necessarily form a nice alpha helix or a beta turn or whatever. They're just very random conformations. And so this makes it really tricky for us to sample from a molecular simulation point of view. But we have techniques such as the Replica-exchange based techniques that enable us to do this. I won't go through this slide now, but the point is that we make extra sure that our conformational sampling is suitable for the fact that we have these intrinsically disordered peptide structures that we need to get a hold on. So on with the goal of our work, we're trying to do something kind of strange. What we really want to do is get something like graphite, a typical kind of 2D material in the bulk, and we want to take something like boron nitride, and then we want to splice them together and maybe make some kind of heterostructure stack. So that was our goal, our ambition, and what we wanted to do was use peptides to do this. I mean, obviously on the top here, I'm showing a picture of someone dealing out some cards, and can imagine trying to do this for 2D materials, right? You can peel them off and you can stick them down in a very manual fashion. But this can really mess up the sheets that you're trying to actually lay down, and they can get a lot of damage and defects as a consequence of that. What we want to do is something that's more like what we see down the bottom, where we can use a more sort of hands-free approach to be able to use peptides to both exfoliate and reorganize these 2D materials in aqueous conditions, and hopefully at temperatures that maybe might be ambient, but will not be too hot. So this was our goal. And so we came up with a concept known as a beam. This is a chimeric molecule, as I talked about before. But in this case, we have in our beam, which is a biomolecular exfoliant and assembling motif, we have a domain at one end that will bind to one type of 2D material, such as molybdisulfide. And at the other end, we have a domain that will bind to another 2D material, such as graphene. And then in the middle, we have a fatty acid chain. And what we've been doing is exploring the construction of these beams to be able to bring these two materials together. And this means that we need selectivity between, say, our molybdisulfide binding domain and our graphene domain here. So we want to make sure that there's not too much crosstalk between the two. The peptide-driven exfoliation. I'm going to skip over the synthesis. The concept of that is pretty simple. So we've already noticed that when we use peptides such as the P1 peptide, which is a known graphene binding peptide, and this was discovered at AFOSR Research Lab in Rajesh Naik's team. This peptide is known to bind to graphene, and we've used that as a way to actually exfoliate graphite into graphene in liquid water. And we're showing you this process here. So we start with the natural graphite. If we actually just sonicate it without adding any peptides, we just get graphite crashing out of solution, and we end up with a clear supernatant here. If we actually add the graphene binding peptide when we sonicate, we're able to make or create colloidally stable solution of peptide, what we assume is peptide decorated graphene sheets. And you can see here the resulting liquid that we end up producing, because clearly got a suspension of graphene in it. So doing this with sonication means that we can keep the temperature down, so it's not too hot, and that we it's not too rough a process, so we anticipate that there's not too many defects there either. And that was really the first work we did there in the graphene space in JFIS Chem C that we published. And that was also a co-public publication with three members of the research team led by Rajesh Naik at AFRL. So I'm going to skip over this synthesis part, but really the idea here is that we're conjugating on the fatty acids, as I told you about. The fatty acid is the part in the middle, and I've just got it. I'm just looking at the chat here. But then what we experimented with that fatty acid linker in the middle is just how long should it be? How many carbons should it have? And so we ranged from 6 to 12 carbons, and we experimented with that. Oop, I don't know why that's not advancing. So I want to show you some simulations. So I've talked a lot about the background. Now we should get into what the simulations are doing. And so what we wanted to do originally is just ask the question, well, okay, so these peptides, they can exfoliate graphene, but how does it do it? And what's really the mechanism? So when you think about exfoliation, there's got to be something that happens at the start and then something that happens at the end that enables this colloidally stable suspension. In the first instance, during the sonication process, we know that the molecules need to be able to help peel off these molecules, the graphene sheets from the stack, right? So they've got to be able to assist in being able to move these molecules off the top of the stack into solution. So that's step one. And then step two, which I'll talk about later, is that once these sheets have been peeled off and are in water, they need to be prevented from reuniting with other peeled off sheets, because they would just clump back together again and crash out of solution. So there's two parts of the process. The first is the actual peeling the sheets off, and the second is stopping them from reuniting once they're free and just bobbing around in solution. So in the first part, we're interested in how do these molecules actually help with this peeling off process. And to do this, we ran a couple of speculative simulations. So under the sonication conditions, we assume that you can make that the graphene stack has a bit of what we call an expanded gap at the top. So this top left-hand corner sort of schematic that I'm showing you, it's got all these gray bars, which is our graphene stack. And we think that under sonication conditions, the topmost graphene sheet, which is this one shown in red, slightly lifts off the stack. So it's a little bit of a gap there already. Now, if you just turn the sonicator off, then that gap just closes immediately. And we did a lot of simulations to demonstrate that that gap closure indeed happens in the absence of peptide, and that it happens pretty quickly. What we also found is that, well, then we started exploring what happens if we put the molecules, like our peptides, into that expanded gap. Will they stay there? Will they stop that top layer from reuniting with the rest of the stack and just kind of closing up? Or will they, do something else? So we did some tests where we inserted our peptides into the gap. And here, if you can see my cursor, where for this pre-inserted setup, we're looking at the stack from above. So we made these hexagonal graphene plates. I'm showing you this one in red but there's a whole bunch of gray ones underneath it. And I've made this red one see-through, so you can see that we inserted our peptides into this gap. And then we run some simulations and we see, well, do these peptides stay here? And the point is they do. They really don't move out. And in fact, that P1 peptide, which we know is a peptide, a graphene binding peptide, will stick onto the graphene very well. So it's absolutely critical. If we put the molecules into that gap, they stay there, right? The gap does not close up. The molecules get all disordered. I mean, this is an initial structure that I'm showing you here. They get all messed up, but the thing is that they don't leave and they don't get squeezed out like toothpaste. So they stay in the gap, and they can stay in the gap for a very long time. So we demonstrated if you put the molecules into the gap, they stay there. So the molecules are having a benefit. And then the next question we asked was, well, if we actually didn't put the molecules in there in the first place, but just put them nearby, would they spontaneously go into the gap themselves? And that's these spontaneous insertion simulations we did, where we put the peptides nearby to the stack. So again, we're looking at it from above. And then we just give the opportunity to actually infiltrate into the gap. And we find that indeed happens. So here, what I'm showing you is a little movie. There's water everywhere, which you can't see, but you can see that these peptides ultimately kind of work their way in. So there were several peptides in this actual movie, but I'm only showing you one because otherwise it looks very messy and confusing. But I've highlighted here in green this tryptophan, which is a really great binder to graphene. And you can see how it gets a little toehold in there and kind of drags the rest of the molecule in. So this is the mechanism that we consistently see for the ingress of these molecules into the gap. So the P1 binding affinity and selectivity is really important for being able to support that initial process of being able to lift off the graphene sheets. And this is just some snapshots of both just P1 on its own, which I've rendered here in blue, but also some P1 with a fatty acid conjugated to the end, which is here in pink. And the thing you can see here down on the bottom right-hand corner is that when we conjugate the fatty acid onto the peptide, we get these really cool wraparound structures. So they wrap around the edges of you can see the red graphene sheet I've highlighted here, the top sheet, that these molecules kind of wrap around the edges. And what we found is that our hypothesis is that protects against defects. And again, the great work that was done at AFRL, led by Rajesh's group with Rahul and Joe, was able to show experimentally that when you have the fatty acid conjugated onto the peptide, you indeed get fewer defects. And we think it's because of this edge wrapping protection mechanism. We've also done some work to further examine what happens if you add the fatty acid to one end of the molecule rather than the other. And we know that experimentally and from our modeling that both support pretty good exfoliation and also very good protection from defects. And here in the top right is actually some of that experimental data that was produced from Rajesh Naik's lab in AFRL, here using the Raman spectroscopy to be able to determine the fraction of defects there. It's really important for us establishing that hypothesis. So I told you about that first process, like the molecules are able to assist with the peeling off. Now I want to tell you a bit about the second process, which is what happens when after all the sheets have been kind of peeled off the stack and they're in solution, we want to stop them from being able to reunite, because if they reunite, they will just make a big kind of, they'll just, what's the word, agglomerate, and then just really crash out of solution. So it's how do we actually maintain a colloidally stable suspension? And here what we did was we tried to understand, actually what I might do is just jump to that. We tried to understand what the free energy was, or we tried to measure the free energy for two peptide decorated graphene sheets, as I'm showing you here in the top left. So this is panel A in the top left. We wanted to take these two sheets and slowly bring them together and calculate the free energy change as we do this. So anything could have happened, right? As you bring these two sheets closer together with this distance, which is indicated as the 5.5 nanometers. So as that goes from 5.5 nanometers down to, something very narrow, like say 2 or 1.5 nanometers, we wanted to know really what that change in free energy was and what was happening to these peptides in the middle. I mean, did they just get squeezed out like toothpaste, or did they stay put? And what we found with our simulations is that when you do bring these together, the peptides indeed do stay put. So what I'm showing you here in this graph on the right-hand side is how the potential amine force is basically the free energy change of our system as we go from, as this red dot indicates, a separation of these two graphene sheets of like 5.5 nanometers. So they're quite far apart and they don't really know about each other yet. And then we gradually bring them closer and closer. So each of these colored dots is associated with a picture here that shows you, well, you know, a typical snapshot of what was going on in the simulation. So here in the blue dot, it's at 4.5 nanometers. The yellow dot, it's at 3.5 nanometers. And you can see they're almost starting to touch at this 3.5 nanometer kind of range. And that's where you start to see the free energy start to really kick up and increase, right? And get quite repulsive. So this is a positive free energy, which tells us it doesn't, you know, the system is not going to do this spontaneously. So you can see that it just starts to increase from that point at the yellow dot, which is where it's 3.5. And these peptide molecules that are on the separate, that are attached to the separate sheets, you see they're just starting to sort of tickle each other, right? They're just sort of coming into contact. By the time we get to a separation of 2.5 nanometers, which is up here in the top right-hand corner, you can see now that these two peptide layers on the graphene sheets are really starting to kind of mesh and get very close. And once they're super close here at 1.5 nanometers, the purple dot, they're almost indistinguishable from each other, right? You've just got a sort of a really thick and compressed peptide layer in the middle. But these peptides do not escape. So in our simulations, we never saw one escape actually from the middle, from that sort of intersheet kind of layer. They stay put. What does escape is all the water. So there's as these two sheets come together and get really close, water gets trapped in this sort of mixture of peptides and water, because it's all being done in water. But ultimately, the peptide stays put and all the water gets squeezed out. And once that happens, there's some parts of this peptide that could really repel each other, right? Because it carries a bit of positive charge and negative charge, and that repulsion starts to get quite extreme because it's a very dehydrated situation in between these two layers, even though the entire thing is dunked in water, right? So there's water everywhere, but it all gets squeezed out. And that's what's driving this really big increase in that repulsive free energy. And it's basically why the peptide does so well at maintaining this colloidally stable suspension. So it's able to tolerate, I mean, if you could, it really doesn't want to get that close. It would certainly never get as close as 1.5 nano unless there was something really applying pressure to it. So this tells us that for these sheets to approach each other, even at all, will just lead to quite an increase in the free energy. So they're so good and stable. So we've done a lot of work on graphene, and what I want to do in the time left remaining in the talk is just tell you about some other 2D materials, because graphene is probably the best studied, and we wanted to move on and investigate some others that are less well studied. And the next one we tackled was hexagonal boron nitride, and finding some peptide sequences that really worked for binding to hexagonal boron nitride was well, it was a bit of a struggle. We did find some that were done by the group of Hanagata and co-workers, and they published these two sequences, BP7 and BP1. And we ended up landing on BP7. It was identified as a very strong binder to boron nitride. And there's a story that goes with this whole bit of experimental work, which I'm not going to labor today, though. I guess that's what this slide was all about. But the take-home message here is that we were able to identify this BP7 sequence and work with that. So for us to be able to model what Mark's experiments were doing, we had a real challenge, because there was, at the time, there was no way, using what we call a force field or an approximation to all those interatomic interactions I spoke about before, there was no real way to be able to do that, there was no force field out there that we could just take off the shelf and start using. We had to create our own. We'd already done that for the graphene work many years before. And so we had to go about and create one for the boron nitride for the first time. So this is to model how water talks to boron nitride and surfaces, and how peptides also interact with boron nitride surfaces. And this was some really tricky work. We used electronic structure theory to be able to determine what those interactions would be for small biomolecular fragments sort of interacting with the boron nitride surface. We had to use Van der Waals DF based density functional theory to do that. And of course, in density functional theory, even with the Van der Waals DF, which is really the Rolls Royce of being able to calculate that sort of weak interaction energy with a surface, we still have reservations. We're still skeptical about whether or not the information we're getting is really accurate. And unfortunately, with boron nitride, there was no experimental evidence out there about the binding energy of a water molecule on the boron nitride surface, which is really surprising. There is a really great benchmark that we've used from the quantum chemistry world, which was a quantum Monte Carlo random phase approximation calculation, very high powered. If you know about electronic structure theory, this is really the creme de la creme. And we used that information to be able to scale our DFT data to get what we thought was an acceptable level of binding. So we scaled all of our DFT data to this one benchmark. So this tells you sometimes when you're creating a force field and you don't have a lot of experimental or very high quality electronic structure theory data to compare with, it can be very challenging. So we're able to calculate a bunch of these interaction energies for different biomolecular fragments on the surface. And in this graph I'm showing in the bottom left-hand panel, this was our fit between our force field and our DFT calculations. Very, very nice fit. So we feel like the force field we created can certainly recover the electronic structure theory data were generated. And then what we used was like one test to be able to see if our force field was on the right track, because again, there's not a lot of experimental data out there for us. And what we had was the boron nitride water contact angle. That was our 1 point of experimental validation. So we did a whole heap of simulations of little water droplets on boron nitride surfaces, and we were able to determine that our force field was on the right track through that. But obviously, it's always better to have more sources of information, sources of truth that you can use. So when we created the boron nitride force field, we were able to calculate the free energy of interaction of all 20 amino acids that are naturally occurring with their interface. I won't go through all the ins and outs of that, but there are some papers there that summarize all of that work. And this gives us a really nice benchmark so we can understand which residues maybe bind more strongly to the boron nitride than others. And so we were able to really take that to the limit in some recent work that we did. It's done by my postdoc Ray Tao here. We just published last year, where we were able to take all the information that we had of all the amino acid binding onto boron nitride, which no one had known about before, right? This is entirely new information. And we took the BP7 peptide, and then we broke it down into these little tripeptides. We calculated the free energy of the tripeptides. So we're trying to break it down into smaller units and get some modular thinking about how could this peptide be rearranged in terms of its tripeptide motifs to perhaps make something that binds more strongly or more weakly. So we're able to do that. But more importantly, we're able to compare with Mark Connect's really great quartz crystal microbalance measurements of these peptides interacting with the boron nitride surface. Now, the original work by Hanagata had done some alanine scanning, where they'd replaced each of the residues in this sequence with alanine, and then they measure the binding again on the surface of the boron nitride. And what they found is that if they modified this tyrosine at position 8, it really reduced the binding. And if they modified some of these other residues, such as this lysine here at position 6, it made hardly any difference, and so on. So this was really intriguing. The one in particular, the the BP7Y8A. So what we found was the residues where this alanine scanning had the most difference and really knocked down the binding were the residues that we predicted were the ones that bound best to the boron nitride surface in the first place, or of the best. And so we went in to kind of investigate what that was. In terms of also which residues also had the least effect when it came to the alanine scanning, they turned out to be the residues that bound the least well from our predictions of our simulations. So again, that's also consistent. And this again gave us great comfort that what we were doing was the right thing in terms of the force field was on the right track. We have since confirmed that those were Hanagata's experiments, which used a fluorescence assay, which sort of has a few question marks over it in terms of being able to establish the binding strength from that. So what Mark was able to do was re-examine those alanine scanning sequences using the quartz crystal microbalance. And from our point of view, we use Replica-exchange to predict the structures at the interface. And again, what we found was really consistent. That in particular, when we modified this tyrosine at position 8 to an alanine, we lost all contact really with the surface. And that was exactly what Mark was able to measure with his experiments. Another really nice thing is that we started attaching fatty acids to this BP7 molecule because we want to make these beams ultimately. And what Mark's experiments found is that for one of the particular fatty acid combinations we use with BP7, we would get this incredible dissipation response when they use their quartz crystal microbalance dissipation measurements. And this dissipation response suggests that these molecules stand upright on the surface and make a very viscoelastic layer. And what we found from our simulations is that for this particular configuration of the molecule, when you have the fatty acid at the C-terminus, you indeed maximize the number of states that kind of stand upright off the surface. So again, that was a great way for us to check in with the experimental results and get a sense that the force field and the approach that we're using was not completely silly. We've since gone on to actually create the beam. So this is the synthesis that was used, and we've been using those to examine how they interact with both boron nitride and graphene. But before I go on with that, I just wanted to tell you very quickly, because I know I don't have much time left, about work we've been doing with molybdenum disulfide. So that was another situation where we needed to create a force field, and because there wasn't one out there in the literature. So again, we used the Van der Waals DFT calculations to calculate a bunch of binding energies. In this case, we did have some experimental data for the binding of thiophene and butene on the surface. And that was really helpful, gave us a great sense that we were on the right track already in terms of our DFT binding interactions. And we just did a ton of work. So we had over almost 350 DFT calculations that were supporting the fitting of this force field. All the work is summarized here. It was done by my postdoc Nan in ChemCom and also in Chemical Science that came out last year. But long story short, we were able to create a force field that was very faithful to not only the binding energy, but also the binding geometry, which is really important. So it's got a high structural fidelity as well as energetic fidelity. Again, we calculated the binding energy of all of our amino acids and got a sense of which ones bind better to molybdisulfide than others. We had some nice experimental data that we were able to compare with that showed that we could predict some structural information that was consistent with what had been measured experimentally. So again, this gave us some confidence that at least the force field wasn't doing something silly. And we've since been doing those exfoliation simulations that I told you about earlier for graphene. We've since done these for molybdisulfide, and that's some work that's currently in preparation and will be coming out real soon. But again, we found that we could explain why some of the molybdisulfide peptides that we've been using were able to help create really nice colloidally stable suspensions. So I'm going to skip over the work I talked about for beam because I think I'm starting to run out of time here, and I know that we need to leave time for questions. I just wanted to talk about one thing that we've done since that project has wrapped. And that is that we've been investigating 2D materials, graphene, molybdenum disulfide, boron nitride. And we wanted to, since then, we've leveraged the work that we've done with that to be able to start modeling really complex graphene oxide substrates. And the reason we got interested in this is that you notice in the literature, everyone's models of 2D materials look perfect. In fact, it's what I call the Instagram version of like a 2D material. So we know that there can be a gap, right, between the Instagram version of something and the real life version of something. And here in this case, we know that in the graphene oxide world, the Instagram version of graphene oxide is what we know as the Lev-Klinovsky model. It is a really perfect looking model, and you've got like basically the chicken wire of your carbon here is just, there's no defects, there's no holes, there's everything sort of so perfect. And you've got this very nice distribution of oxygen in terms of the hydroxyls and the epoxies. But what happens in real life, of course, is you've got raggedy edges and holes, and like the oxygens bunch all up in one place, and then there's not so much in other areas. And these are the kinds of things that we want to try and capture. So we wanted to come up with an algorithm, a sort of, well, actually, it's a software package that you can actually use. It's called HireGo, and it will create these very complex graphene oxide structures for you. So we've got a little algorithm here. I won't go into all the ins and outs of it, but the point, long story short, is that we're able to introduce defects, we're able to introduce holes, we're able to introduce heterogeneous sort of oxygen distribution, and we're able to combine all of those to make some really complex looking structures that are more like what you would expect in a real geo model. When you're trying to model, for example, permeation properties, if you're thinking about water desalination, or even as supercapacitors, these kinds of defects can really matter. And that's what we've been doing at the moment, is they're really leveraging off that work that we've been doing in the 2D materials to be able to do some more practical simulations using these geo sheets, which are really complicated and kind of horrible looking, but also more like what we'd expect geo to really be like. So just to wrap up, I think I've just gone on a little bit too long, but hopefully we've still got some time for questions. So what we've done is really being able to forge ahead and combine, most powerfully, Mark's experiments with our simulations. It's really pushed us from the simulation side. We had to come up with force fields. We had to do a lot of tests along the way, but and there's still a lot of things to do. For example, I never talked about boron nitride exfoliation, and that's because we know already that peptides can't exfoliate boron nitride, and we've been investigating why. But because these are public release slides, I'd like to be able to go into that, but that work is still not published, and so we're a bit nervous about sharing that with the rest of the world. We are also wanting to explore how the graphene oxide structures we've made can be interfaced with biomolecules, and of course, generating our graphene, for example, boron nitride heterostructures using our beams. So it just remains for me to make many thanks, because without everyone here on this slide, we couldn't have got any of this work done. And most importantly, that we are supported by the AFOSR and also the researchers at AFRL in this work. During the time that we held this grant on the 2D materials, we had quite a few program managers. So starting with Hugh, and then Aura, and then Pat Bradshaw, all of these people we thank for their support of this project. And at AFRL, it was Rajesh and Joe and Rahul made such critical contributions to the work that we were doing. As I mentioned before, our supercomputers here in Australia have been an incredible resource that have enabled us to do all the work that I told you about. And last but not least, Professor Mark Knecht and his team at the University of Miami, who we've been working with and who've provided this critical complement to our simulations. And thank you all for listening today. Thanks heaps, and happy to take some questions.
Okay, thank you very much, Professor Walsh. We have about 4 or 5 minutes remaining for questions. And so at this point, I have one question in the Q&A. For those participants who want to ask a question, there is a Q&A button down at the bottom of your screen. So if you have a question, you can click that and type it in there. So we have a question from Charles Rumberger. What group are you working with at AFRL is question one. And number two, do you see potential applications of biomolecules for self-assembled patterned metasurfaces?
All right. Well, yeah, as I've mentioned, the people that we on this particular project that we worked with was Rajesh's team. So that was Rahul and Joe are really critical in being able to do some of the experiments, in particular the characterization experiments. So that for this particular project, those were the people we worked with at AFRL. In the other projects that I work on, as Todd already mentioned at the start, this was really the human performance team. So Jorge and Peter Moreau, very critical for the work we're doing. So those are the groups that we interact with at AFRL. Also for question two, that's a really good idea. And in some ways that's easier than trying to get these sheets to independently organize, but to template them onto a surface and to make metasurfaces is something that we certainly have given thought to. We thought it would be a nice stepping stone to the final goal of being able to make these 3D heterostructures that could just assemble in solution easier to give a support first and to sort of start building that templating. But to use that to also make these metasurfaces, yeah, it's a very cool idea. Thank you for that.
So I was curious about the experimental work. You know, you said in many cases there's just not a lot out there. So what would be your sort of top 1 or 2 experiments on your wish list? Like if you had some particular experiments that you would like to see some results from.
Yeah, that's such a great question, Todd. And I think the answer is here in the chat. So Peter has very kindly pointed out that some new NMR experiments, and for us, NMR is just it's really the Rolls Royce of experimental data, because you can make a direct connection between the simulations and the NMR data. This is why it's so precious to us and very powerful. So Peter, thank you for that suggestion. The dynamic nuclear, the DMP NMR, I think would be very, very helpful in this area. Also for us would be some more measurements of smaller molecules adsorbed onto, like just being able to measure the free energy of interaction of these small molecules onto certain surfaces. The real key there is being able to make sure that we can match up what we're doing in our simulations and being able to keep the molecules small. So try peptides, or even, you know, maybe no more than 5 residues. We can get quite an accurate estimate of what that binding free energy is. But it's being able to bring together a free energy measurement and I think some kind of NMR observation that gives you the structural information at the same time. And combining that with simulation is perhaps the most powerful thing that we could do. So that's right up there on my wish list.
Oh, great. Thank you very much. Dr. Roach, you have your hand raised.
I do, because I cannot use the Q&A because I'm a panelist. So I have to verbalize my question. I'm sorry. I was going to try that, but it didn't seem like it would work. So my question has to do with, you know, the work that you presented is really intensive in terms of beginning with 2D structures and really trying to understand how to model that and then compare that model to actual data that tells you that you've got it close or right on target for the functionality once you've grown that, once you've done that. Can you talk to the complexity of going to the next level of creating 3D structures and then trying to test that? Because what you've convinced me of is how difficult it is to get to the one part, and I know you alluded to this, but how difficult in your mind is it to get to the next level of what you're trying to do three-dimensionally over?
Yeah, that's a really great point, Pat. And I think you're right, it is complex, but in some ways, what I've really told you about this, this afternoon, is basically the fundamental work that we know then gives us confidence that what we can model subsequently is not complete garbage, right? Is not entirely silly. And that's really important. And maybe it sounds like I'm denigrating my own work, but I'm not. As someone who does modeling, it's very important to stay skeptical and to make sure that we're always trying to make touch points with experiment to make sure that we're modeling the right thing. Now, when it comes to these modeling these 3D structures and that sort of stack assembly, it is rather more complicated, but we can sort of build this up step by step, because in much the same way that the experiments have been done. So to explain, our thinking is that and I didn't talk about this in the slides again because they're public release and the work's not quite published yet, is that we use the beams, the beam molecule itself, the hybrid molecule, the chimeric molecule, actually is a great exfoliant, say for graphene. And we can then use that beam to exfoliate graphene, in which case, and we've already done both simulations and experiment to establish that when the beam itself is on the graphene sheet, that the part that should be attaching, say, to the boron nitride actually does hang off the surface and presents itself so that it's able to interact then with a boron nitride sheet. So in the experiment, we exfoliate with the beam first with the graphene, and then we can dial in pre-exfoliated boron nitride. And by using a little bit of BP7 in our pre-exfoliated, well, no, not BP7, we actually use another molecule. It's called tannic acid, which I'd love to, we have a lot of simulation data on the tannic acid and how it works for the exfoliation of boron nitride, but it's not published yet, and we're just a bit reluctant to share it today because the slides are going to be out there. But we know already that we can use that as a sort of pre-exfoliant, and then the beam can then actually displace the tannic acid. So we can kind of build it up step by step experimentally, and we can do the same thing in our simulations. So we can construct models, and we have, of the graphene sheets with the beam molecule stuck onto them and decorated onto them, and then have a similar boron nitride sheet approach that and be able to demonstrate that they can indeed sort of start to kind of get together and make those sandwich structures that I showed you before. The next step after that is being able to explore on a much bigger length scale how these sheets then start sort of systematically organizing into larger stacks. And for that, we need to simplify our model a little bit more so that we can model many more particles in our system to be able to demonstrate that organization on a much greater length scale. So that would be our next challenge. I think we couldn't do that last step if we're modeling the system at the atomic level. It's just too much. The number of atoms in our simulation cell would just kill us, and we wouldn't be able to get to the timescales we need, I think, to be able to cover that. But it is possible with a little bit more work to simplify those interactions that I talked about before.
Thank you. I don't see any more hands raised or questions in the Q&A. Cindy, I think we're about out of time. So I would like to thank our speaker, Dr. Walsh, once again. And, we're excited. Like I said earlier, this grant is only a few months, the current grant is only a few months old. So we're looking forward to some, continued work with Professor Walsh. And mostly, really excited about the strong collaboration with AFRL. So big thanks to the AFRL team that is, just participating and collaborating and making use of this research. And with that, I'll turn it back over to Cindy.
Thank you all for joining us today, and I will be ending our session in the recording. And again, it will be published to our YouTube channel once we have the public release. Thank you all for attending, and good night.
Thank you. Goodbye.